Hello students, welcome to grade 12. Before we start today's lesson, let's recap and revise what we have seen in the previous lessons. In the previous lessons, we have seen population and population growth and models of population growth. Lastly, we have seen human population growth. Today we are going to see the first lesson of unit 3, genetics, genetic crosses. And we are going to see Mendelian inheritance and other non-Mendelian inheritances. Now, let's start today's lesson by an activity. Students try to relate chromosomes, genes, alleles, and characteristics of organisms. What's the relationship between chromosome, gene, allele, and characteristics of an organism? In grade 11, we have seen the definition of these terminologies. So relate chromosome, gene, allele, and characteristics of an organism. Well done. Let's again revise and see the meaning of each term. Chromosome is long strands of DNA wrapped around protein called histone. The role of chromosome is to carry genetic information in the form of genes. So genes are carried by chromosome. Chromosomes are located in the nucleus of eukaryotic cells. Human beings have 46 chromosomes, 23 from mother and 23 from father. Genes are a specific segment of DNA on a chromosome. So genes are carried by a chromosome that codes a particular protein or functioning molecule. The role is to determine traits by encoding the instruction for the development and function of an organism. So chromosome is to carry genetic information, whereas genes are determining the trait by itself. A gene on a chromosome may determine eye color and other traits. So eye color is one trait, one characteristic. Allele are different version of the same gene. One gene could have two alleles. So these are versions of a gene. They can lead to variations in the expression of a trait. So when traits are expressed, alleles lead to variation. They could be dominant allele or recessive allele. So dominant allele are alleles that has expressed themselves, and recessive alleles are expressed only when two copies are present. Recessive alleles are alleles that are dominated by the dominant allele. So capital A symbolizes the dominant allele, which is homozygous. In the heterozygous case, capital A, small a. When there is a dominant allele, capital letter is used. For recessive allele, small a, small a. So this is homozygous, same allele, which is recessive. Characteristic are observable traits or features of an organism, such as eye color, height, longness or shortness, blood type. We have seen two types of blood grouping, ABO blood group and Rh positive and Rh negative. These are characteristics. The relation to genes and alleles characteristics are determined by the combination of allele of an organism. So alleles determine characteristics. For example, the combination of alleles for the gene determining eye color in blue, brown, and green comes from the alleles. So the alleles determine characteristics such as eye color. The interconnection chromosomes carry, as we have seen, genes. So they are the house for genes. Each gene copies a specific position or locus on a chromosome. So genes are carried by chromosome. They do have specific site that's known as locus. Alleles are variations of gene that lead to differences in characteristics. So gene has two alleles. These two alleles are determine and lead to variations in characteristics. Characteristics are traits or observable characteristics. So the combination of alleles inherited from parents determine an organism's observable traits. These observable traits are characteristics. So chromosomes, genes, and alleles are all in all to determine an organism's or offspring's trait, observable trait. So this trait first starts from chromosome, which is found in DNA, and genes are carried by chromosome. Genes have alleles. These alleles could be either dominant or recessive. Homologous pairs are the chromosomes in a eukaryotic cell usually come in a pair called homologous pair. Chromosomes are usually in a pair, and this, when they are in the usually come in a pair, they are known as homologous pair. In each chromosome in the homologous pair have corresponding gene that together determine the same trait. Locus in a polar loci is a position of particular gene on a chromosome. So genes are found on chromosome. The position of a particular gene is known as locus. We have seen also what's homozygous and what's heterozygous in grade 11, but let's revise again. Homozygous are an organism, or homozygous is for a particular gene if it has the same allele. Homo means the same. For each chromosome in the homologous pair, if the pair of genes are the same, then they are called homozygous allele. If they are different, hetero means different. An organism is heterozygous for a particular gene if the gene has different allele. For the gene on each of the chromosome in the relevant homologous pair, they are found in a pair. If this pair, one of them is different from the other, then it's known as heterozygous allele or heterozygous gene. Now let's see the difference between genotypic and phenotypic. Genotype describes the pair of alleles for a particular gene possessed by organism. Exactly, you put the genes that are found. Whereas in the phenotypic case, describes the trait or characteristic determined by a particular genotype. Now let's proceed and see what is monohybrid cross. Mono means one. So this is an experiment that involves only one trait. only one trait. You remember Mendelian experiment? And pea plant, he have taken only one trait. This could be either dominant or recessive traits. And Mendel have used Punnett square to predict the outcome. To remind you again, these are the characters, 7 characters and 14 traits that Mendel have used. Flower color, the dominant trait was purple and the recessive trait was white. And this is the F2 generation ratio. Flower position, axial and terminal. Seed color, yellow was dominant and green was recessive. Seed shape, round and wrinkled. Pod, constricted and inflated. Pod color, green, yellow. Stem length, tall and dwarf or short. These are the 7 characters and 14 traits, and their ratio. Let's see the explanation of Mendel. Mendel have said that every trait like flower color or seed shape or seed color, these characters are controlled by 2 heritable factors. These are now called genes. So every character is controlled by heritable character known as genes. They can take different forms of allele. They can be dominant or recessive. They can be homozygous or heterozygous. If 2 alleles in an individual are different, then one is dominant and will be expressed in the organism's appearance or physiology. This is known as dominant. And one is recessive, cannot express unless the individual has 2 copies of the recessive allele, and that's in that case it's known as recessive. Dominant trait is a trait that masks the appearance of recessive traits. So the one that masks the other trait is known as dominant and the other is known as recessive. Mendel also explained the only physical link between the generation in the gamete or sex cell. This must pass genes from one generation to the next generation. So genes are used to transfer genetic information from one generation to the other generation by using gametes and sex cells. The heritable factors or alleles separate when the gamete sex cells are formed. Each gamete therefore contains only one allele controlling traits. This is Mendel's law of segregation. You know, Mendel has 3 laws: law of segregation, law of independent assortment, and the law of dominance. One of the laws he used was law of segregation. He also stated that the gamete sex cells fuse randomly at fertilization. Now let's see how parents showing one version of future have children with the other version. Now let's take an example, earlobe. So let's take an example of genes of earlobe. It has 2 alleles, free earlobe and attached earlobe. These are the 2 alleles. Everyone has 2 alleles of this feature in all their cells except the sex cell. A person could have either of these 3 scenarios: 2 attached earlobe per cell, that is homozygous for the recessive allele, or 2 free earlobe alleles per cell, homozygous for the dominant, or one attached allele and one free earlobe allele per cell. This is heterozygous case. This one is homozygous for the recessive. This one is homozygous for the dominant, and this one is heterozygous. Heterozygous. This figure shows that the outcome if both parents had free earlobe with the genotype. So you put capital E, small e, capital E, small e. Then in the first box you get two capital E's. 1/4 of the boxes will be capital E, and the second box capital E, small e, and the third capital E, small e again, and in the fourth and the last two small e's, small e from here and small e from here. So 1/4 would become homozygous recessive, one become dominant, and the two others are heterozygous. So we would expect 3 out of 4 children to have free earlobe. This phenotypically, genotypically they are different, but phenotypically they do have free earlobe, because for free earlobe capital E is simple. So in both the 3 boxes, 1, 2, 3, we do have capital E. So 3 children from the 4 children would have free earlobe. Whereas one of the children would have attached earlobe, small e. So phenotypically the ratio would be 3:1 ratio. This is phenotypically. In other way, if we make this expression in percentile, 75% or 3/4 of probability that any particular child will have free earlobe, 75% of the children will have free earlobe, and 25 or 1/4 of probability would have attached earlobe. But remember, genotypically, the ratio would be 1 to 2 to 1. One dominant allele, 2 heterozygous allele, and 1 recessive homozygous allele. So this is genotypically, but phenotypically they have 3:1. 1, 2, 3, capital E is for free earlobe. Now let's see and proceed to test cross. Test cross is a way of breeding between recessive phenotype with the dominant phenotype. Recessive breeding between recessive phenotype with the dominant phenotype. The dominant phenotype zygosity can easily be identified by this test. All the test cross are considered to be back cross. So all test crosses are back cross, but all back crosses are not test crosses. Back cross is a breeding of F1 generation, hybrid with that of any 2 parents. If F1 breeds with the homozygous dominant, due to offspring becoming 100% dominant phenotype. How can we tell if an allele is dominant or recessive? This is an example. Squares show males and circles show females. Shaded symbols are affected, have widow's peak, and W dominant allele for widow peak, and small w recessive allele for no widow's peak. The horizontal line is symbol for marriage, and the vertical lines show parents or children. So here we have the W capital W, small w, small w, small w, and small w, capital W, capital W. And the result will become, at the end of the cross, the result will become WW or W small w and small w small w. This is recessive, this is dominant. This is first generation, this is second generation. The third generation, we do have this widowed dominant allele and the recessive allele. The hybrid cross is another inheritance which involves two traits. We have seen that monohybrid cross involves only one trait, but this one involves two traits simultaneously. And in this, the hybrid cross law of independent assortment works, and at the end of this, the hybrid cross, we have phenotypic ratio 9:3 to 3:1 ratio. This is the result of the hybrid cross of Mendel's hybrid cross. So now you can consider that the parent is having round yellow seed and wrinkled green seed. Here we do have shape and also size. And the result would be 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. So 9:3:3:1 ratio. Let's try to answer this question. Are alleles always simply dominant or recessive? The short answer is no. Sometimes alleles could be either codominant or incomplete dominance. Codominant is the two alleles of the gene are both equally expressing themselves and being dominant. In the heterozygous, both are expressed. So this is known as codominant. And this kind of codominant is found in flower color of snapdragon, and the result will become pink, where the parents were red and white. So pink color is not white, it's not red, it's a mixture of red and white. So pink color is codominant. Both parents express themselves. Are there always two alleles? The answer is again no, because we do have triple or multiple allele inheritance. And the example of multiple allele inheritance is ABO blood group. Genetic roles are applied in predicting offspring of genotype and phenotype using back cross and test cross for analysis. This brings us to the end of today's lesson. We wind up today's lesson here until we meet in the next lesson. Goodbye.